Hello, everyone. Welcome back. I'm going to make sure I have my color for my drawing. So we're going to get right into what happens in meiosis and how it brings about unique characteristics. So fertilization occurs when sperm and egg come together. So you take two sex cells, one sperm and one egg, and you produce an offspring that has the full number of chromosomes but has unique characteristics from both parents. So basically you're taking haploid 1, so half the DNA, plus haploid 2, the other half of the DNA, producing diploid right here that is unique. So how do we get this uniqueness? The reason that we sexually reproduce is because of the ever-changing world. So if we are perfect, if we fit into the environment perfectly, then why would we want to change what we already have, right? If I am perfect, I'm not going to change to fit in better. I'm already perfect for the environment. But because the environment is ever changing, we have to change with it so that we can produce offspring that are better suited to the new environmental pressures. And that's what sexual reproduction does. It gives us the ability to produce offspring that are better suited to the environment. And so there are two major ways that this occurs. That is through this process called crossing over and this process called independent assortment. So what is crossing over? Crossing over occurs in prophase 1 of meiosis 1. And it occurs between the non-sister chromatids of homologous chromosomes. So here we have one chromatid, and here we have another chromatid, right? So we have a chromatid from the blue chromosome and a chromatid from the red chromosome. So what happens is the chromosomes cross over, and so here you see crossing over on the long leg of the chromosomes of the non-sister chromatids. Crossing over can also occur up here. So you can have crossing over up here, you can have crossing over down here. You can also have no crossing over occur. So for every chromosome that undergoes, or for every cell that undergoes meiosis, the chromosomes, homologous chromosomes, may or may not cross over. And if they do cross over, they may cross over once, they may cross over more than once. And so this increases genetic diversity. So we have 23 pairs of chromosomes, which means we could produce, we could have crossing over occur on every single chromosome. One time, we could have crossing over occur 2 times on every single chromosome. Or we may have no crossing over occur at all. Or, and what's more likely, is that crossing over will occur on some of the chromosomes. On some it might occur more than once, and on some it might not occur at all. So that increases our genetic diversity, because we end up with chromosomes that have new information. So this chromosome now, this blue chromosome here, is different from this blue chromosome here, even though they are the same, they came from the same individual because of crossing over. The other thing that increases unique characteristics or increases genetic diversity is independent assortment. So independent assortment basically means that chromosomes line up independently of all other chromosomes. So homologs will always line up together at the metaphyseal plate. This occurs at metaphyseal plate in metaphase 1, okay? And you might get, in this case, the small blue and the small red lining up, blue on the left, red on the right. And then the big blue and the big red also line up, blue on the left, red on the right. That could occur. What could also occur is that the red big one lines up under the blue, and the blue big one lines up under the red. So in both cases, we are going to produce cells that are different. So over here we have blue and blue together, red and red together. Over here we have a little blue and a big red, and a little red and a big blue. So this can occur. Now again, we have 23 chromosomes pairs. So we could get all of the blue chromosomes. So all of dad's chromosomes in one cell, all of mom's chromosomes in another cell. We could get all but one of dad's chromosomes or all but one of mom's chromosomes. So they line up independently of all the other chromosomes at the metaphyseal plate in meiosis 1. And because of that, we get unique groupings of those chromosomes, and that is independent assortment. This was seen by and discovered or described by Gregor Mendel, which we'll talk about in chapter 23. So actually, you know what? I'm going to stop here, and I'll get into meiosis versus mitosis in the next video. So I'll talk to you guys in a little bit. Bye.